Welcome to the Talking Precision Medicine podcast, brought to you by Genialis. I'm your host, Raphael Rosengarten, CEO and co-founder of Genialis. On this podcast, we discuss the future of healthcare and health technology, and how advances in data and data science are fueling the next industrial revolution. Please subscribe to the show on Spotify, Apple Podcasts, or wherever you listen to podcasts. And for more information on Genialis and our unique approach to AI-driven precision medicine, please visit our website at www.genialis.com. That's www.genialis.com. Now, on to the show. Hey everybody, welcome back to Talking Precision Medicine. It's my pleasure to welcome my friend Tom Neyarapally. Tom is the founder and CEO of a really exciting new AI for drug discovery company called Archetype Therapeutics. Tom, welcome to the show. Tell us a little about yourself and tell us about Archetype.
Thanks so much for having me. I'm Tom. I am the CEO of Archetype Therapeutics. We're an AI native drug discovery company founded a few years ago. Our whole premise is a patient-first approach to drug discovery, in which we start with patient data, both clinical and genomic data from patients, and build patient genomic classifiers. Think about things like Cologuard or Oncotype DX or Decipher. These are tests that measure the activity of a patient's genes in disease tissue and associate that with clinical outcomes and build tests. And the purpose of those tests are to predict outcomes for patients, which helps design care paths and other types of decisions that patients and their physicians make. We use those types of classifiers, but we use them for a different purpose. We use them to determine whether drugs will be effective for patients with that given disease. And the way that we do that is we use generative AI, which is, of course, a hot topic these days. Our application of generative AI is to be able to learn the relationship between the structure of a drug and what it's going to do to the biology of a disease. So we then combine those two things. So we build what we call these generative chemogenomic models that allow us to understand, as I said, the relationship between the structure of a proposed drug and its effect on biology. And then we use these patient-derived classifiers to understand the relationship between biology in a set of patients and clinical outcomes. So when you put that sandwich together, you can go from new drug or proposed new drug to the biology to the outcome in a patient, and we can screen at massive scale. So we're talking about screening 1 billion molecules that look like potential drugs in a day, understanding what they do to biology, and then understanding in turn what those changes in biology might mean for clinical outcomes in that patient cohort. And then we generate hypotheses. The hypotheses are that the drugs that look interesting in that paradigm that I just described are potential new drugs. And then we bring them through the same types of in vitro and in vivo and other types of validation that are typically done in the drug discovery process.
This is fascinating, and I love this approach, partly because, it's so complementary to what we do at Genialis, where we build genomic, or in our case, transcriptomic patient classifiers focused on omics and clinical data. But one thing I want to clarify, because I don't know that everyone's going to get it, you're essentially designing drugs, your Gen AI is designing drugs against what you call biologies. How is that different from how other approaches to either rational drug design or Gen AI, or even traditional screening works? What do you mean by biology?
Yeah, so you've just articulated the two comparator paths to what we do. So one would be a path that's target focused. Think about, I have a target, maybe AlphaFold has given me the structure of a target, and I'm trying to optimize a structure against that target. I'm trying to optimize the three-dimensional structure of a small molecule to make sure it fits in a binding pocket of a protein that's going to knock that protein out of the action in the game. That is not what we're doing. And there are really cool companies that do that. That, of course, presupposes that they have the right target and that target is going to solve the problem. So if that is the right target and you only need to hit that target, then that approach is great. And that's not the problem we're trying to solve. Then you also alluded to the other side of it, which is massive high throughput screening. That is an approach where, again, you can take a target or some kind of biological assay and you can run lots of drugs against it and see what actually works in that assay, what affects the biology in a way that you want in that assay. Now, once again, that is interesting and that is valuable, but that doesn't scale up to billions of molecules. It would take more time and money than the world's ever seen to be able to actually screen billions or say even trillions of drug-like potential drugs using that type of approach. So both of those two approaches you mentioned are valid. They're used in drug discovery. They have been for years, and they're both improving in scale, but neither of them can really touch the combination of, and I like to talk about this, the different aspects of how what we're doing is a little bit different than those approaches and other approaches. It's a combination of biological relevance, which is starting from patient data. That's super important, we think. Let's start with real patient data from cohorts that we're trying to solve a problem for. It's the scale that we talked about. So being able to screen at the scale of billions. There are companies that can make on demand molecules from these libraries of billions to trillions of molecules, companies like Enamine and eMolecules. Let's use all of that chemistry space. Not everybody can do that. We can. So that's the scale. And then there's things like speed, which a traditional preclinical drug discovery process takes years. We've been able to do this in months. And then cost. If it takes only a few months to do, it's not going to cost the perhaps $5-10 million or more that it might take in a traditional preclinical drug discovery program. So those are all things that are different than these approaches that you mentioned.
Yeah, I think it's interesting because a lot of people will talk about sort of the AI enablement of new chemistry as giving us a chance to have a lot more shots on goal. But what I like about this approach, since you're thinking about how do we find a chemistry that perturbs a biology rather than a specific a priori defined target, you're also making the goal a lot bigger. So it's a lot more shots, but on a much wider goal. And intuitively, if the tech works the way you say it does, that means you're a lot more likely to find chemistry that's going to actually modulate the disease the way you want it to.
Yeah, exactly right. I would say a wider goal, and I would argue a more relevant goal. So a lot of these cancer drugs, and by the way, I should clarify, our platform is not limited in its application to cancer or really any disease area. It's really about being able to get the disease biology measured. Where is the action in the disease? Can we get to that tissue? So if you think about CNS and neuro type applications, the challenge might be, can you get to the right tissue in the brain where the disease is playing out? But that aside, the idea that the biology is not just about one target, but that there are multiple pathways in play with respect to how the disease plays out is important. And by not focusing just on a target at the outset, we can cover that. Now, on the other hand, one of the objections I've heard, and that's perhaps a misinterpretation of what we're proposing, is this idea that, oh, well, you're ignoring targets and the drugs have targets. Well, we're not ignoring the targets. Of course, physically these molecules bind to things. This isn't magic. The drugs have targets, but they may not be the targets that you believe they are. We've found this. So, for example, in some of the public binding data that's available, that's sitting in NIH and other libraries, we'll see that somebody's registered that molecule A hits target B and C, but then when we go in the lab, it doesn't seem to actually hit B and C. So the reproducibility of the belief with respect to what the targets are may also not be true. So we don't have to fall victim to that. We can cast a much wider net. So as you said, the goal's wider. The goal might be more relevant as well when we're trying to find drugs that are going to address the problem that the patient has. The trade-off there is the causality that and I like to analogize this to system one and system two, what Daniel Kahneman talks about, where you have to trade off precision for how much data you bring in. So system one part of our brain allows a lot of data in and gives you an answer. It's that gut feeling of, hey, this is a sketchy person, or this is somebody that I want to spend time with, for example. We do that with data. We let more data in, we let more genes in, but the answer may not tell you exactly where the causality is. What is the target? Fine, that's okay. We can then use system two, which is the other part of our brain or the other part of our scientific processing in this case, which is that calculating, reduce it down to a few variables, get the scientist on it. Well, we do that as well. So that's the part where we want to say, hey, we found a hit. There's a molecule from this billion set that we've run that's interesting. Then we have expert scientists. We've built a stellar SAB, for example, world-class people. They help us understand, well, what's actually going on with that drug? What is it really doing? We're not throwing that part out, but we're saying, let's cast a wider net first. And that goes back to your wider goal point.
Yeah. So I have two questions then. The first one, you have a patient classifier. You think you have the therapeutically relevant or biologically relevant, we'll call it target biology, the biology you have to perturb. You run your generative AI, you now have a molecule. How do you figure out what that molecule actually does bind with? Like, where does that happen? And then the second thing is, do you expect it to be a single target, or is there a chance that actually polypharmacy, in other words, promiscuous binding is in fact the reason why it works or works better?
Yeah, great. Both are good questions. So with respect to the first question, there in one use of the platform, there's an a priori hypothesis about the target because we pour into the top of the funnel molecules that have purportedly known targets. So that's one way to use the platform is take every drug that's ever been to clinical trials, for example, and you can repurpose drugs to your heart's content that way. That, of course, doesn't work if you take an enemy library of 38 billion molecules that no one's ever synthesized. So therefore, no one knows what they bind to because no one's ever physically made them. In that case, once we've shown in silico and then in some in vitro, if not in vivo assays that the molecule's interesting, then we're going to run through standard assays. There are different types of assays you can do to try to see what the molecule sticks to, for example. And this is outside of my particular realm of expertise as a chemical engineer and not a PhD biologist, but there are tried and true approaches to go fishing for what is the target. But the advantage we have is now already have a hypothesis that this molecule is going to move the expression patterns in the right way. Now we can go fishing for what the target is, but we've narrowed the field quite a bit from starting from 38 billion molecules. So that answers that first question. And then remind me your second question, related question was.
The second piece was around the idea of polypharmacy. Like, are you trying to optimize for single target specificity,
Yes.
or are you okay with the idea that it could hit a bunch of things in a pathway, for example?
Very much okay with and like the idea that we might have one molecule or perhaps even a combination of molecules that might press on different aspects of that gene expression pattern in a way that moves the whole pattern towards wellness in the patient as judged by the classifier. So it could be either one molecule that has polypharmacy inherent in it, it's a promiscuous kinase binder, or it is multiple molecules that we layer in because they play a different tune each. And so we end up with this melody where one is playing one aspect of the piano, one's playing another aspect of the piano because it's pressing on different expression levels and different genes. So it's both of those things are valid and interesting to us and are possible with what we do.
This strikes me as a very different approach than we're used to hearing about, talking about, reading about. Undoubtedly, you get some raised eyebrows when you talk about this with people, especially those who've been in the industry a long time. What kind of evidence do you have to show them that, hey, this might work or this does work? I remember seeing some stuff you guys put out at AACR that was drawing a crowd.
Yeah. The question has loomed from the very beginning. Interestingly, the feedback, even from the big pharma folks that I've gotten, is hey, cool, very different approach. Like the idea. Early on, 3 years ago, the feedback was go prove it. This is a cool idea. Go prove it. First level of proof we had was the ability to predict the outcomes of clinical trials before they occurred. And a really interesting example of that is in PARP inhibition in prostate cancer. So in metastatic castration-resistant prostate cancer, PARP inhibitors were believed to work in patients that had DNA damage repair mutations, like a BRCA mutation. And there was a class of PARPs that already existed by the fall of 22 that did exactly that. Pfizer then was developing another PARP inhibitor called talazoparib. And our predictions back in 2022 was that this PARP inhibitor, because of its structure, remember, our platform just takes the structure, no other information, and says it's going to move the expression of important genes in prostate cancer in this particular way. And that in turn means something about outcomes in this patient cohort. Now, our patient cohort, by luck, happened to be a patient cohort that was not enriched for DNA damage repair mutation positive patients. So the hypothesis would be PARP inhibitors shouldn't work in this patient cohort. And so when we ran all the PARP inhibitors, the ones that already existed on the market, as well as this drug in clinical development from Pfizer called talazoparib, what we saw is the other PARPs didn't work in the cohort in silico, and Pfizer's drug did. So when we showed this to some of our expert friends in prostate cancer, they said, hey, you've had some great predictions with this platform, but this one seems to be wrong because we know that PARP inhibitors don't really work in this patient cohort. And I said, okay, well, I guess we'll have to wait and see what happens when they read out with their phase 3 results. And then at ASCO in 23, the readout was actually this drug does work in this patient cohort. And there's been additional studies, and this is a drug of great interest in the industry right now. So that was our first level of proof. Showed that to folks, and they said, that's cool, but that's not your drug. Show me something where you discover something yourself from your own biology with drugs that you might have an IP claim to. And at that time, we were fortunate. We both, Paul, my co-founder and I, had been in the industry long enough And we happen to be former colleagues of a guy by the name of Charles Powell at Mount Sinai. He's the CEO of the Respiratory Institute there, so he's a senior leader there, but his research interest is in early stage lung adenocarcinoma. And he had just published in the summer of 2022, I believe, in Nature Communications, a paper on repurposing drugs in early stage lung adenocarcinoma. And I thought, wow, this is a perfect wine pairing because we have an engine to generate drug hypotheses, and he has all the wet lab assays published in the top tier journal to be able to prove what we have. So we're a wet lab agnostic shop. In fact, we don't have a wet lab, so we work with wet lab partners. And so my proposition to Cap, as he goes by his initials, was, "Hey Cap, why don't we generate some hypotheses, and then your colleagues can test these in your lab?" And he said, "Cool, let's go for it." So we launched that last summer. We took only about a month to build computational models to make predictions, to identify drugs that were interesting. And then we could compare them, not only run them in his assays, but compare them against the best stuff that they could discover in the lab over the previous decade. They had used the stuff that Paul and I have done for a long time, which is systems and network biology approaches. And so they used a network biology model. They discovered key targets, and they found a drug that hit those targets called AMG 900. There's an aurora kinase inhibitor, and they thought aurora kinase inhibition would be great to head off early stage lung cancer. So that was what we were going up against, was a published paper in a top tier journal, a lot of research, very smart people. And we have this new approach with a little bit of proof. And that's what we went into last summer. We generated hypotheses both with respect to completely novel molecules as well as clinical stage molecules. And we focused the first part of our analysis on clinical stage molecules. And we found that MEK inhibition, the inhibition of MEK 1 and 2, might be interesting. Now, remember, we don't search for targets, we search for drugs, but the targets can come along for the ride if the drugs have known targets. So that's what happened here, is that we found that mirdametinib happens to be really effective in our in silico model to head off early stage lung cancer. But then also when we went into CAP's in vitro and ultimately in vivo models, including a GEM model that is exquisitely engineered to have the lungs of these mice develop invasive lung cancer, that our drug, in comparison to the best that they could find, this Amgen drug, had a material difference in heading off invasive lung cancer that you could see in the pathology of the mice. And then in addition, the mice lived for 40 weeks compared to a control mice. So they lived a long time. They had favorable pathology when we did the sacrifice of the mice at an intermediate time point. So just this really exquisite data. And that was strong enough that Cap and his team, to their credit, said, "Hey, this actually worked. This is really incredible." And they submitted an AACR abstract over the winter months going into this year, and it was accepted. And there was an invited oral talk at a mini symposia in our very first submission as a company. First time we ever submitted anything, we had a talk, not a poster, at AACR. And I've been told, I'm not an expert in this, but I've been told that that's actually a pretty big deal. So we were very proud of that. And that was what was presented essentially. Now, what I didn't cover is we then took some of that learning and we said, well, if this mirdametinib, if this drug is interesting, what about molecules that generate similar gene expression profiles to mirdametinib, but that are proprietary, that haven't been to the market yet or haven't been in the clinic yet? And we identified those molecules from billions. And those also worked when we went into the in vitro assays. And then finally, we took one of those molecules and said, can we engineer even more activity into these molecules just by making molecules that we think will induce better gene expression patterns? So no wet lab data to create the hypothesis. And we did. We engineered in better activity in silico. We sent those proposed new molecules to India to be synthesized. They got shipped back, just the best stuff, the quickest stuff they could make from a list we gave them. And lo and behold, those molecules engineered in greater in vitro activity. So we have this whole continuum of work in lung cancer that started with, will this work at all? Okay. Can we repurpose existing drugs? Can we identify novel molecules that look like those existing drugs? And that whole path kind of has played out accurately, that this stuff actually works. And it's been a phenomenal journey over the last year to have that real wet lab proof that comes from a out-of-tier lab that's not our lab and so on.
Hey there. I hope you're enjoying this episode of Talking Precision Medicine, powered by Genialis. We'll be back in just a moment. I recently got back from the Bio International Convention, where the biggest takeaway wasn't about drug discovery, it was about regulation. The FDA is moving fast to bring artificial intelligence into the review process, and that could change how we approach all of clinical development. I wrote up some reflections on this short LinkedIn article. Just search for my name, Raphael Rosengarten, or head over to the Genialis page to check it out. And while you're there, give us a follow. We're always sharing updates at the intersection of biomarkers, AI, and drug development. Now, back to the conversation. I love that. And you said some things that I want to kind of use to transition. For better for us, our listeners got dunked in the deep end of AI drug discovery. 'Cause Tom and I like to geek out on this sort of stuff. But you had mentioned that you and your co-founder Paul come from a world of systems biology and kind of have been in the industry a long time. I want to learn a little bit more about how you came to start this company, but maybe what you were doing kind of before that. Notable mark on your CV was chief commercial officer and a member of the founding team at Semaphore. For nearly half a decade, or more than half a decade. Tell us a little bit about, as you said, not a biologist, not a life scientist by academic training, right? So how do you even get into all of this, and what's that journey been like?
Yeah, perhaps I'll start before Semaphore. It'll explain how I got there. Really, if I rewind the tape quickly to the beginning, my dad was a lifer at Pfizer, 30 years at Pfizer as an organic chemist. And so, but he was on the manufacturing side. So in his world, the chemical engineers ran the show. So guess what I did in undergrad? I was a chemical engineer at UConn on a scholarship. After that, I was briefly in management consulting, working with pharma. And then I went to grad school at Cornell and I met these seemingly crazy physics grad students that wanted to apply math and powerful computing to cancer biology, which we're talking about this, and there's probably lots of people talking about this today. But back then, 25 years ago, that was a very novel and maybe crazy idea that you could model biology exquisitely using computation and math. And they started a company, these friends of mine that I met in 2000, called Gene Network Sciences, then became GNS Healthcare. It's now called ATIA. Company still exists in a different form up in Boston. Colin Hill and Aya Khalil are the two friends I mentioned, and Colin is still the CEO over there. They started with mechanistic modeling of cancer biology, so building up biology by pathways. What are the known pathways? Can we turn that into math? And can we simulate the pathways to understand what happens when we intervene in different places? Within the first few years, they changed to something that was completely novel at the time in terms of the scale of doing it, in parallel with some great work that had been done by others. So the parallel work I'll talk about first, that was Eric Schott and others who did this idea, built this whole approach to inferring biological networks from data. And they published some seminal papers, he and others, when they were at Rosetta Informatics back in the 2000 timeframe. And shortly thereafter, that company was bought by Merck and they continued that work. And so Eric is a pioneer in that type of work. Eric was the chair of the SAB of this little company, Gene Network Sciences. And what Gene Network Sciences was able to do, and Paul McDonough, my co-founder at Archetype, that's where I met him 19 years ago at Gene Network Sciences, where he and I, along with Colin Aya and Bruce Church and a couple others, were co-founders and co-inventors of the core technology, this Bayesian inferential approach to learn biological networks using these massive supercomputers. Again, maybe more mainstream today, but completely crazy and out there 20 years ago, and proved that this type of approach could work. So that was my foray into this, was meeting these grad students 25 years ago. And then after 4 years as a lawyer in New York, saying, I'd much rather be in these cool startups than be a partner in a law firm, and took a big pay cut, moved to Boston, joined the GNS crew, and spent 9 years with them. So really, even then, way ahead of its time, even in 2015, 10 years ago, when I left, and I left to join Eric at what was then Mount Sinai and the genetics department. But the idea was to spin out the genetic testing lab that was captive within the genomics department to create a company. At the time it was called Mount Sinai Genomics, but the name that we selected was Semaphore. So I was the first outside management hire outside of the academic department to do that, and hired as chief commercial officer. What was interesting about that company is that the vision was to build these massive clinicogenomic data sets to do the type of stuff that Genialis does, to do the stuff that we want to do at Archetype. But the way to do it, there was the selling of genetic tests. And this might sound familiar because there are other companies now like Tempus and so on that do that. The vision then was, we already had this great carrier screening test for people planning on having children. We planned on having cancer testing. And the idea was when you sell the test, you have an opportunity to interact with patients and providers, and you can consent the patients to share their data to be able to build drugs for patients like them. That was the concept behind Semaphore. But what was complicated about that is those are two very different businesses, a health information business and a genetic testing business. Now, I think people have figured this out. And again, I give Tempus as an example. They have this great data set and they've been able to figure some of this stuff out. It's not easy because investors expect different things of a health information business than they do of a genetic testing business. There's different skill sets that are required to go sell tests. So, for example, when Eric hired me, I thought that the idea was that I would go sell these health information deals and the stuff that I was doing at GNS. And it was actually the mandate was go sell those tests because that's the way we're going to get the data. I had never even taken a genetic test. So I had to figure out what are these genetic tests? How do they work? And I had to build a national sales team. I visited humbly, hundreds of doctors' offices where I had to carry the food in alongside the reps and get to know the doctors. And it was an amazing experience. I had no experience in sales as opposed to business development, but learned it. We figured it out. We built a national sales team and built a couple $100 million in revenue a year. And we went public on the back of that, combined with Eric's vision of the health information business, which is really why I and many others signed up for that. So it was an amazing journey. I got to ring the bell at NASDAQ and be on TV for a few minutes on CNBC in the morning in July of 2021, 5 years after I started there. And then I left soon after that. One of the things I learned about that whole business is of going public is that you end up raising a lot of money and then you have to do something with that money. And you can't go hire $500 million worth of people. That would have been cool. Or buy $500 million worth of data. That would have been cool. Inevitably, you have to buy another company. The company they bought was GeneDx. And I won't comment too much on what happened afterwards, other than that the surviving company is GeneDx, and people can read about the story of what happened there. But I left before all of that played out. So I was the first guy in. I was also the first management team hire to leave. I wanted to move on to the next thing. I, in fact, moved to Puerto Rico briefly, consulted in the private equity world for a big New York City private equity firm looking at precision medicine strategies. That also gave Paul and I space. I pulled Paul into that project. That gave us space to start this company. So Paul's had these phenomenal ideas for a long time. Again, he goes all the way back to Rosetta Informatics 25 years ago. And he had this idea that I've just shared with you about Archetype. So that's my path from GNS through Semaphore all the way to Archetype.
No, it's absolutely fascinating. I mean, it's, just as a brief history lesson for listeners, this notion of AI for drug discovery, it's not new. It's just the words we use are new. And frankly, the mathematics we're using today are extraordinarily new. I mean, the advances in the actual science of artificial intelligence has been insane. But a lot of the concepts and the problems that people are trying to solve, as Tom's described, he's been working at this for 2.5 decades now. And it's one of these examples of every overnight success took decades to build towards, right? So I want to know a little bit more about your vision then of Archetype. Do you envision this becoming its own biotech? Like, do you want to be a biotech? Do you want to be a platform company? And a number of years ago, when venture capital was cheap and pouring into the industry, there were a lot of platform companies spinning out portfolios of joint ventures. Do you envision being kind of a tech platform that partners? I mean, how do you engage the market right now?
Yeah, it's a question we get often, and we can look now. Now there is a history of companies known as quote unquote AI drug discovery companies that goes back about 10 years. To your point, ML and other approaches have been used for this going back another 10, 15 years. But let's say it's been about 10 years that this term started getting used. So you can look at Recursion as one of the earliest companies that gets named frequently in this. And then there's a cadre of other companies that have come along since then. All of them have been asked this question by investors and others. And all of them essentially, in one way or another, have towed the line between the two. So they've built platforms. Most of them have real interesting AI platforms that do different things. And they've had to prove that the platforms work. The way you do that is not by just pontificating. You've got to discover drugs, and those drugs ultimately have to get to the clinic and be successful in the clinic. That's the true proof of whether these approaches work or not. Now, as a very small company that's just pre-seed funded, we can't get to the clinic, but we can create molecules and discoveries that can get towards the clinic or through the clinic, if not through the clinic. And so our path up through now has been We think we have a really valuable and generalizable platform that can be used. It's been used in prostate cancer, been used in lung cancer, successfully in both. We think there's application outside of cancer, so on and so forth. Uh, However, in order for anybody, whether it's in a platform play or otherwise, to really buy into the broader use of this thing, or even the discoveries from it, we've got to take some things forward to a certain extent. So we've spent our effort in validating that the platform works, and that's some of the data that you heard throughout this discussion. And then on top of that, advancing either on our own or through partners some of those molecules into the clinic. So that's been our dogged focus over the last couple of years. And then we think the answers will reveal themselves to some extent as to, well, what do we want to do with that? Is it easier for this platform to be put in the hands of many folks in a tech-enabled service type of way? Or is there something really special about the model we built in lung cancer where it just keeps churning out gold that we just need to keep running with that? A version of that is you spin those out. You take the lung cancer effort, you spin that out. You take the prostate cancer, as you prove it, you spin it out. And that's what you were alluding to. And there are platforms that have been built in big venture capital firms that have done exactly that with some level of success.
Absolutely. You see all of the above, right? You see companies that keep all their molecules for themselves. You see companies that partner. I think of in silico medicine, of all the AIDD companies, has the molecule that is furthest along in the clinic of anybody, right? They've got a phase 2 IPF molecule that we're all rooting for so that we can celebrate the success of the method. You have even companies like Nimbus, which are like hub and spoke model biotechs, right? So I can take your point that there are a lot of possible structures and they're not mutually exclusive.
That's right.
And then on the biology side, because ultimately your secret sauce is a combination of the approach plus your generative, well, call it generative chemistry platform. This idea of perturbing biology, I'd love to hear a little more about how you think about finding partners on that side. And just, the caveat is, Genialis and Archetype have sketched out some really exciting collaboration opportunities because again, what we do is build those patient classifiers. But, how do you evaluate what makes an interesting patient classifier?
Yeah, we're looking for a couple of things. One is, what patient cohort has that patient classifier been built from? We think that that's critically important. And does that align with an unmet medical need? And has it been validated? Those are really the things. And that was the attraction in building the partnership with your team, is that that's the work you all have done over a long period of time. You've gone and validated various classifiers that are very relevant. I think of KRAS as just one example. These classifiers are answering questions that are immediate and relevant for drug discovery. And they're a perfect wine pairing for a tool that can then take those very classifiers and tell you what new molecules will do in those classifiers and then create putative new therapeutics. So that's the nice wine pairing. There are others that do things like this. And then there are people that don't do what you do, but they might generate the raw data that could be turned into this type of thing. So those are the people that are interesting to us. In that latter case, then we or others have to bring the skill into turning that raw material into interesting classifiers. Of course, the advantage working with you all is you've already done that, so we don't have to do both steps. We can just start with the classifier that already works. But those are the types of partners that we're looking for.
I mean, the good news sandwiched with the bad news is that, yeah, we've gotten very good at Genialis at building classifiers that can stratify responders and non-responders for, cancers that are still seeking, good therapies. That's the good news. We're good at it. The bad news is we still see a predominance of non-responders, right? If you think about KRAS as an example, I think, the two approved molecules each have efficacy rates in non-small cell lung cancer of roughly, upper 30s, low 40%. So that means 60% of the patients getting those drugs are not showing benefit and therefore still need something better, right? So that's the bad news, is that our best targeted therapies, or a lot of them are still, important tools in the toolbox, important arrows in the quiver, but they're not panacea. And so there's still very high unmet need. And I guess the good news sandwich to that is, if we can confidently identify non-responder populations and hand off to a team like yours, we move the needle towards true precision medicine, where we have drugs designed for specific, in this case, not specific N of one patient cohorts, but we'll call them biotypes or phenotypes of patients who still very much need a new medicine.
That's right. I get this question all the time is, are you doing personalized medicine? And people have this idea that personalized medicine means an N of one. Totally not true. Personalized medicine exists on a continuum between everybody in the world all the way down to that N of one. And there's value along that path. This idea that cohort size could be 50 people, for example, that have they failed 5 drugs. They have various other covariates that are associated with them. So that's pretty close to an N of one out of several billion people on the planet, and perhaps good enough to find a really exquisite drug for that type of person. Doesn't have to be an individual. So that's the way we look at it, is patterns of gene expression in a cohort that we want to solve a problem for. That's really the personalized medicine aspect.
Let me zoom out and maybe step sideways a little bit and just ask, in general, in the rapidly changing world of drug discovery, of precision medicine, of diagnostics, just kind of the whole continuum from, well, I guess it used to be target to patient, but now this continuum that we're trying to close the loop on. What else is really exciting to you? What do you see coming out of AACR and ASCO and going into bio? Like, what are the industry trends that give you the most hope and kind of stamina in the morning to get up and do this sort of thing?
Yeah. I mean, one thing I'll talk about is we focus on gene expression just because that was our starting point with the platform. But this type of approach, we already have ideas sketched out how to bring multi-omic data into the mix. These are just finer and finer grain measurements of what's actually happening in the biology. There's a temporal aspect. Some people ask me about this, and we're able to do this better. There's a spatial aspect. So, what are the omic modalities? What is the time and space in which those things play out? There's all this cool stuff happening in that world, all of which can be brought into this type of paradigm. It'll get more and more complicated, but the AI power is increasing every day. We can talk about the singularity of where are we on that elbow of the curve, where thinking about it linearly may not be the right way to think about what 1 year from now looks like. So I'm excited about that, both on the computation side and then the richness of the data that is becoming available day by day. And then we don't have delusions of grandeur. It's not going to all happen in archetype, but we think that approaches like ours, combined with approaches like yours and the new data modalities that are coming out, are going to accelerate drug discovery. Now, of course, I've always felt that was true, but I think that there's this convergence of these things that's happening in a much more material way now than, say, 5 years ago.
Yeah, really great observations, and I share a lot of the enthusiasm for the same reasons. Kind of two questions to wrap things up. One of them is just looking again back at your career. You know, you gave up what I guess could have been a very lucrative but stuffy job as a corporate lawyer, in New York, and decided to build companies for a living, right? And they happen to be companies that have great missions and work towards sort of the betterment of human health. But now having, contributed to building out GNS, which, as you mentioned, still exists as Aetia, and so it's a very durable company, truly sort of the original gangsters of this kind of, um, tech-enabled service modeling of biology, and then Semaphore, which was a very rapid rise to a unicorn and a public company, and now with Archetype. To anyone who's just getting started or starting again, what are some advice, some pitfalls, some just general, words of wisdom that you've learned over, again, the last 20 something years of doing this?
Yeah, first of all, for me and why I'm doing this, one of the mission, and this is also true for Paul, is I've lost loved ones and people close to me to cancer young. One woman in her 30s and another woman in her 40s, close friends from growing up in southeastern Connecticut. So for me, it's pretty easy to be motivated every day because I was in this industry well before my first friend even got sick, and yet we couldn't do enough for those people. So the idea is that the tools are actually mature now. So this is the moment in time now, as opposed to 5, 10, 15, or even 20 years ago, because I was doing this in all of those prior time points, that we can actually make a difference. So part of it is your mission to, be a partner in a law firm, or a banker, or is your mission to solve problems for patients every day? And if that's what you're really passionate about, I think it's an incredibly exciting time. Probably a much better time now, despite the financial challenges and the unique macro environment at this point in history that we're facing. The tools, the technology, the data, the people, the passion is still all out there to do it. So first thing is I would say don't be dissuaded by the fact that some of these things, macro things, are a little bit more challenging right now. Because if you're passionate about this, people are going to make things happen. I still think we're going to be super successful. I think many other companies are going to be as well. But I think that, to me, that's the biggest thing. The other thing is, don't assume that because you spent a bunch of time training for one path that you have to do that. So for me, I'm still paying, I'm 50 this year. I have another several years of paying student loans. At 26, I remember, but I'm paying until 56. That's now scary. It's a little bit sooner than I expected that 56 will sneak up on me, but I'm paying for that school from Cornell, law degree and an MBA there, 4 years and a lot of debt. But very valuable training, but I didn't feel stuck as a fourth year mid-level associate that I had to keep doing that. And nobody should feel that way. I feel like that's a loss. Now, there's nothing wrong with going down that path if that's what you're passionate about and that's what you want to do. But people that feel like, hey, I'm in a big company, I put my time in, I got to VP or I got to director or whatever the level is, I can't give up on that. I'm going to lose my place in the ladder. I would totally dissuade people from thinking that way. And if you want to take the jump, take the jump. I think it's more fun over here on this side of the pool, but that's just, I'm biased, obviously.
The ride certainly goes up and down with
It
a
sure does.
greater frequency.
That's right.
All right, last question. I try to ask everyone this. If we all do our jobs right, if you guys do your job right at Archetype and achieve the success, 10 years from now, what does medicine, what does cancer care look like? What's changed?
Yeah, I think the biggest thing is we're going to cover, we probably maybe won't have everything cured and solved. Much more of rendering cancer a chronic disease that gets attacked with lots of strategies that evolve over time, and that there's an information layer behind that that informs maybe it's Genialis 5.0 at that point, 10 years from now, where it's, well, at any point in the evolution, we're going to be able to go back to the model and say, this is the right intervention. And that intervention might evolve pretty frequently rather than, hey, somebody's on drug A for a year, then they're on drug B, then B fails, and then we're out. So I think that that's what's going to be different is the speed, the nimbleness, and the precision towards one patient that we're going to have these arrows in our quiver. We're going to have lots of arrows and they're going to be more precise. However, I think it's not necessarily the case that we'll have cures. People talk about cure as shorthand. I don't know if that's necessarily true for lots of cancers. Doesn't have to be, though. If we render cancer more of a chronic, treatable, doesn't affect quality of life so much, then that's a huge win from my perspective. And I think we'll get there in 10 years for sure.
Well, I share your vision. I share your hope. Tom Neyarapally, thank you very much for your time. Appreciate it.
Thanks so much.
This has been another episode of the Talking Precision Medicine podcast, brought to you by Genialis. Hopefully by now you've subscribed to the show, but if not, go ahead and click that subscribe button and leave us a review. And please do pay us a visit at www.genialis.com. That's www.genialis.com to learn more about how AI is transforming precision medicine. Thanks for listening, and see you next time.